Hello all, welcome to this lesson on microscopy, the third in the series on cell biology. This lesson is tailored to the AQA, OCR and Edexcel GCSE specification, but also suitable for all secondary science curriculums. In this lesson, we will describe the functions of a microscope, to describe how to prepare and observe a slide under a microscope, and to calculate the magnification of a microscope. Now we look at the types of microscope. A microscope is an instrument which is used to magnify objects which cannot be seen with the naked eye. The two main types of microscope, the light or optical microscope and the electron microscope. Now we compare these microscopes based on different properties. The light microscope uses light waves to observe objects. The electron microscope uses beams of electrons. The light microscope can observe both live and dead cells. The electron microscope can only observe dead cells. The light microscope is portable. The electron microscope is bulky. The light microscope is affordable, few thousands of pounds, but the electron microscope is expensive, hundreds of thousands of pounds. The one needs training, does not need training to use a light microscope, but one needs training to use electron microscope. The light microscope has a low magnification. The electron microscope has a high magnification. It can make the image about 1,000,000 times bigger. The light microscope has a low resolution, but the electron microscope has a high resolution. Now we look at the parts of the microscope and their functions. Start off by looking at the eyepiece lens, and you come to the coarse focusing knob, the fine focusing knob, the objective lenses, the stage, and the illuminator. The eyepiece lens, you look through it to observe the image. The objective lens magnifies the image. The coarse focusing knob moves the stage up and down to bring the image into focus. The fine focusing knob makes small adjustment to the stage to bring the image into sharp focus. The stage is where the slide is placed for observation. The illuminator produces light to illuminate the specimen, and the diaphragm found right below the stage controls the amount of light that goes onto the specimen. Now we look at calculating magnification, but before that we look at the definition of magnification and resolution. Magnification is the number of times an image is made bigger. Resolution is how clear an image is, or the ability to distinguish between 2 points on the image. Magnification is calculated by dividing the image size by the actual size. To manipulate this formula, you could use a formula triangle. You have the image size at the top, the magnification at the bottom left, and the actual size at the bottom right. To be able to do these calculations, we need to know a few conversions. We need to understand that 1 millimeter is equal to 1,000 micrometer, and 1 centimeter is equal to 10,000 micrometer. Now we look at a few examples. The first one says calculate the magnification of a 5 micrometer bacteria, which is the size of 2 millimeters when viewed under a microscope. Now, because the units are not the same, we need to make sure we convert them to the same unit. So we convert a 2 millimeter to micrometers by multiplying by 1,000. So we get 2,000 micrometers. We then go ahead and divide the image size by the actual size, so 2,000 over 5, and that gives us times 400. So that's the magnification. We come to the second example. It's asking us to calculate the actual size of a bacteria with the size of 0.4 millimeters when viewed under a microscope with a magnification of times 250. Now we need to change the 0.4 millimeters, the image size, to micrometers, so we multiply by 1,000, and that gives us 400 micrometers. Now from the formula triangle, if we cover actual size, we will get image size over magnification. So we divide 400 by 250, and that gives us 1.6 micrometers. Here we look at the practical skills, and this is a required practical for a number of the exam boards: preparing and observing a tissue under a microscope. So here we specifically look at preparing an onion slide. Now, To prepare an onion slide, you use tweezers to peel off a layer of the onion tissue and lay it flat on a slide. You add few drops of iodine to stain the organelles so you can see them clearly under the microscope, and then you gently place a cover slip over the tissue, making sure no air bubbles are caught in it. To observe the slide under the microscope, you will place the slide on the stage and hold it in place with a stage clip. Position the lowest magnification objective over the slide and move the stage close as possible to the objective lens. You turn on the light source, which is the illuminator. Look through the eyepiece, turn the coarse focusing knob to move the stage down until the image is in focus. Turn the fine focusing knob to make small adjustments to the stage until the image is in sharp focus. You can then use a higher magnification objective to increase the size of the image. To summarize this lesson, let's do a true or false quiz. Take a few moments to make your decision. The resolution of an image is the ability to distinguish between 2 points on it. This is true. Electron microscopes have higher resolution than light microscope. This is true. Fine focusing knob magnifies the image. That is false. That is the job of the objective lens. The diaphragm controls the amount of light that goes onto the specimen. That is true. 0.005 micrometers is equal to 50 millimeters. That is false. That will be equal to 5 millimeters. The formula for calculating magnification is equal to image size divided by actual size. That is true. When observing onion slide, iodine is added to the tissue to make it colorful. That is false. It's to stain the organelles. When observing a slide, the highest magnification objective is used first. That is false. It's the lowest magnification objective. A light microscope can observe both dead and live cells. That is true. And a slide is placed on the illuminator to observe it under the microscope. That is false. It is placed on the stage. See you in the next lesson.